Recording will begin now. Thank you everyone for being here today with Women in Aerospace Medicine, members all over the world, and Dr. Don Kernagis from the Department of Neurosurgery. Neurosurgery, why do we have a neurosurgeon running amok in Women in Aerospace Medicine? Well, we do have some neurosurgeons in the audience of our membership, but Dr. Kernagis is here for a very special reason. She's an underwater explorer and a dive medicine researcher. She's in the Women Divers Hall of Fame and a fellow of the Explorers Club, which I was just inducted into and I'm very excited about. She's going to tell you all about how undersea research translates into outer space research. We're going to be going undersea to get to outer space. The recording has begun. As a reminder, please feel free to leave your cameras on, mute your mics, and drop your questions in the chat. I'd now like to hand the floor over to Dr. Kernagis. Thank you for joining us today, Dawn.
Awesome. Okay, let me get to my screen here. Can you all see that okay? It looks like it's up and running. I have these like the admit buttons, so I'll just keep trying to close those out as they come through. So sorry about that. Anyway, it's so fantastic to meet everybody. Shanna, thank you so much for the invitation to speak. This is really exciting, and just learning more about what you all do too. I think this is an incredible organization. So I'm honored to speak with you guys today. So why neurosurgery and then undersea medicine and also aerospace medicine? So essentially my big interest is looking at ways to protect the brain and nervous system in high physiological stress conditions. So that could be undersea, that could be outer space, but that could also be patients. If you think about stressful conditions for the brain and nervous system, undergoing a neurosurgical procedure is probably one of the most stressful conditions you could think of for the brain and nervous system. And also things like radiation therapy, chemotherapy. So we look at ways to protect patients ahead of time and optimize their brain and optimize the nervous system so it can handle those stresses of neurosurgery or different clinical procedures the best way possible. And so there are a lot of things that we learn across those different populations. So we learn a lot from those patient populations that we can translate over to the extreme environmental populations. So again, as individuals working from undersea to outer space, and there's a lot we can learn from those healthy humans who choose to put themselves in those stressful conditions, and ways that we can protect them in those environments and translate back to those clinical populations that we work with. So kind of goes back and forth across those two populations, which makes it extra rewarding, I think, for the work we do. So I'm going to talk a little bit about how I got into the work that I do, and then talk about just a high level, a couple of examples of some of the research that we're doing with respect to protecting the brain and nervous system. And these are, again, projects that we're working on both with respect to undersea operators and then things that we're translating over to the space populations or even the aerospace populations. And then I'm going to also talk about an opportunity that I had that really blended my first career as an underwater explorer and my current career as a scientist, where I got to actually physically work undersea to translate things to get to outer space. And that was working as a crew member with the Nemo 21 expedition back in 2016. So that's NASA's underwater operation. This picture is actually a picture from Nemo. And so this is actually astronaut Megan MacArthur. She's getting ready to come out for an EVA for a simulated spacewalk out of the habitat. I love this picture. It looks like just so powerful. This, incredible aquanaut going out to do her EVA, and just there's a lot of great energy behind it. It was a really exciting mission to work on. So moving through, I have nothing to disclose. So talk about these different populations that I work with, and you all are pretty familiar with this, but anyway, anywhere from individuals who are working undersea, so especially Navy divers, we've done a lot of work with the Navy diving populations, individuals who are working at high altitude, so that includes pilots. We've done a fair bit of work with pilots, especially out of Naval Air Station Pensacola. And then we also have done a fair bit of work with individuals who are working at high altitude and mountainous environments, for example, so special operators who work in mountainous settings. And then also we do a fair bit of work with NASA and spaceflight crew. So how I got into what I do. So this is actually a picture looking from the entrance of an underwater cave out to the surface. So you can kind of see the trees here. And these underwater cave systems, in particular the ones in Florida, tend to be filled with spring water. So the water is crystal clear. It's gin clear. It's essentially like sitting in air. It's so crystal clear. Just absolutely beautiful cave systems. And so I got involved in cave diving when I was actually 16 years old. I started doing a lot of cave diving, and about 2 years in, after doing a lot of cave diving, I got into underwater cave exploration. And so I worked with projects all around the world, but I eventually landed on working with a project called the Woodville Karst Plain Project. And this is an underwater cave exploration project that's focused in Tallahassee, Florida. Really quick, I'm just going to move this so I can see. There we go, because I think you guys can probably see all of my screen right now. So sorry about that. There we go. So that way you guys can see this. So this is, so if you're familiar with Florida, essentially the Woodville Karst Plain Project is located just south of Tallahassee, Florida, kind of on this eastern side of the panhandle of Florida. And it's absolutely filled with underwater caves, sinkholes. And a lot of these are really, really deep caves. So for those of you who are familiar with scuba diving, typically a recreational scuba dive, max depth is about 120 feet. These caves typically run anywhere from 200 to 300 feet in depth. So the dives that we were doing were extremely high stress, long decompression procedures for those dives because of the depth and also the time at depth that was required to do underwater exploration in these caves. So with the Woodville Karst Plain Project, okay, I don't know why I can't forward that. There we go. So this is just a map of the Woodville Karst Plain. And so these are some of the different underwater cave systems. And the goal of this project that I was working on for such a long period of time, and I worked with them for over a decade and a half, by the way, was to connect these underwater cave systems and also to map them out so we had a better understanding of the karst, the hydrology, the hydrogeology that was in that area, and also where water was flowing underneath the surface of the ground. And that was really important for making building decisions and infrastructure decisions in the panhandle of Florida. We also conducted biology experiments, all kinds of different things to kind of maximize the science that we could do while we were doing these extreme dives. To note, it was a volunteer-based project, so people would come in on the weekends, anywhere from usually a weekend to 2 weekends a month. They would drive down to Tallahassee, Florida, and they would pour all their resources into doing these incredible dives. Average in-water time for a diver was 15 hours. And again, depth of about 200, 300 feet. So a good portion of that was decompression. So trying to slowly come up from these really deep dives to minimize the risk of decompression sickness or the bends. The longest dive that we did was a world record breaking dive, is 26.5 hours, including decompression. And that was a dive where essentially after we connected two major cave systems, they did a swim through dive of 7 miles from one cave entrance out to the other cave entrance to demonstrate that those two cave systems were connected. So just a lot of effort went into these dives. And the thing that got me into research was watching my teammates pushing the limits physiologically, psychologically, and even with respect to equipment when they were doing these dives, and trying to figure out ways that we could best protect our divers before they went into these really, really high stress dives. And this is just an example of what diving in one of these underwater cave systems looks like. Again, gin clear water, absolutely beautiful. But as these divers are going through, keep in mind that they're diving at a depth of 300 feet or around 100 meters in depth. They're doing these dives, trying to monitor a lot of different things. They're tracking how much oxygen they're consuming, how much gas they have left, not just they have left, but also how much does their buddy have remaining, what their decompression procedure is going to look like after the dive. In a cave like this, you're also paying attention to where you're at in the cave, so you don't run into the cave and damage your equipment. Obviously, damaging the cave is a bad thing too. There's a line that runs to these caves that takes you all the way. So essentially, if everything fails and you have to find your way back out, that line, if you can see it kind of in the lower left corner of the video, that line essentially will bring you all the way back out to the surface if you follow it. So you don't want to damage that line. You don't want to break that line with your scooters, for example. So a lot of different things that you're tracking in these dives. And as these divers are doing these dives, they also have a number of factors that are working against them with respect to their cognitive capacity and also their physical abilities. And that includes, I don't know why I can't forward these, I apologize. So that includes a lot of different things that impact you when you're scuba diving physiologically. So immersion itself, actually being immersed in the water, has an impact. It's been shown to have an impact, a negative impact on cognitive performance and also physical performance. So this whole time when they're doing these really long dives, just being immersed itself is actually having an impact on their ability to perform. This risk of decompression sickness or the bends I talked about, inert gas narcosis. So there's a risk of having narcotic effects based on the gas you're breathing, especially if you're breathing nitrogen at depth. And so these divers are breathing different types of gas mixes to try to minimize that narcotic effect. oxygen toxicity. So doing these really long dives and really deep dives, you're breathing high pressure oxygen for an extended period of time, especially during decompression. So you have a risk of not only pulmonary oxygen toxicity, where you have damage to the lungs and you have kind of a effect on your respiratory abilities, but you also have a risk of developing what's called central nervous system oxygen toxicity. And this can be deadly for divers in the water, because essentially could eventually lead to seizures in the water, which is not ideal if somebody goes into a seizure at depth. And so that's something that we're always tracking and trying to manage, that level of oxygen that you're breathing throughout a dive. Carbon dioxide retention because of the equipment, and also increased work of breathing. And I say hypo and hyperthermia. It doesn't have to be to the extremes of hypo or hyperthermia. It could just be that you're cold or you're really hot. And those two things both impact your performance, but they also impact your safety. So they impact things like your decompression risk, your decompression sickness risk, or your oxygen toxicity risk. So that's something that you're always trying to manage throughout a dive as well, and can impact your overall ability to perform and also your safety during a dive. So I got out of, I started kind of transitioning to doing a little bit more work outside of just the diving community. So I transitioned from being an exploration diver. I went to grad school. I was doing work that was funded by the Office of Naval Research, an undersea medicine program that was focused on divers. And then because of that work, I also started doing work in more of the aerospace community as well. And this is actually work that was published by one of my colleagues, Dr. Jeff Phillips, who's at the Institute for Human and Machine Cognition. And he does a lot of work with aviator performance and high altitude performance. And obviously with aerospace medicine, one of the biggest considerations is hypoxia. And so I don't have to tell this group the implications of hypoxia, but I really thought that this study was interesting, where he looked at pilot performance at simulated altitude. So it's 18,500 feet of altitude. And these are trained pilots, so they had hypoxic exposure experience. And he had them on a reduced oxygen breathing device simulating 18,500 feet of altitude for either 30 minutes or until their blood oxygen saturation dropped to below 50%. And then they were allowed to recover. And then they went through and started doing, they continued to do cognitive testing out at 30 minutes, 60 minutes, 120 minutes, and 24 hours. And so, as you would expect, we saw deficits in certain aspects of cognitive performance during hypoxia, and even acutely after hypoxia. But what was really interesting is that certain things like choice reaction time and simple reaction time actually were impacted all the way out to 2 hours after that exposure, even after they had been back on room air. So the thought's always been, okay, you're hypoxic, or the thought previously had been, you're hypoxic, you get back on room air, you should be good to go. But there is this residual deficit in performance in these individuals that there's still a lot of work being done to try to figure out why that is and what does that window look like before return to duty, for example, for pilots. So really interesting work. And so, by the way, hypoxia can affect divers too if their breathing equipment isn't delivering adequate oxygen. So it's kind of that transition from the undersea medicine over to the aerospace medicine world. And then obviously when it comes to space medicine, there are a lot of different things that can impact astronauts. The things highlighted in green are the ones that are considered kind of higher risk considerations, especially for longer duration missions, things that we need to tackle before we think about those really long duration missions. Spaceflight associated neuroocular syndrome. There are a lot of questions as to why these changes to the brain and the eyes happen in space, but we think that there could be some effect of fluid shifts. And by the way, you see fluid shifts in divers, and that's one of those reasons that immersion itself can impact diver performance. Cardiovascular deconditioning, some of the radiation stuff, not so much diver related, but things like immune dysfunction and even behavioral health for the undersea medicine community, especially for a submarine community, those things translate back and forth. And there's a lot of shared research that's being done between the space community and the undersea medicine community to figure out like what can the two groups learn from each other when it comes to some of these different conditions. And so as I think about performance, or as my team thinks about performance optimization and resilience, I kind of think about the work we do as neuroprotection or neuroresilience. We think about these kind of three different phases if you're thinking about protecting an individual in whatever extreme environment or extreme condition they're going to experience. So there's a readiness piece, so preparing the body, preparing the brain and the nervous system to handle those stresses as it comes before they come on board. Resilience, so the body can actually handle those stresses as they're occurring, and then obviously recovery. And a lot of these times, these different phases where we look at ways to protect these individuals kind of overlap with each other, right? It's not just a one-off. If you're looking at a nutrition strategy, for example, that's going to impact both readiness and resilience and actually recovery too. So there's kind of this cycle, but we're always kind of thinking about performance optimization and resilience optimization in these three phases. So I'm going to talk about two areas of research that we've been focused on when it comes to neuroprotection or neuroresilience for individuals in extreme environments. So the first one is optimizing metabolic efficiency and protection. And so this is where normally I would have a show of hands for individuals, but for those of you who are familiar, and I know you all are familiar with ketosis because you're medical professionals, but typically in a lay audience, I say, who's heard of the ketogenic diet, right? It's been all the rage. I think even some of the Kardashians are doing it now. So it's all over the place. But essentially, as you all know, ketogenic diet or fasting can put you into ketosis. And so the state of ketosis leads to the development of production of ketones that your body can use as an alternative fuel source compared to glucose, which is typically what we burn. And there are a lot of benefits that have been shown, especially in preclinical models, for burning ketones versus glucose. So examples of that are simpler transport and breakdown compared to glucose, greater inherent energy per carbon and beta hydroxybutyrate or ketones versus glucose, and then also greater mitochondrial efficiency. And there's also newer research that's pointing towards, even with respect to the brain, there's an increase in production of growth factors and neurotrophic factors that are protective and help with recovery, in particular after a stressful exposure for the brain and the nervous system when you have ketones being metabolized versus glucose. So how you get into ketosis, again, endogenous ketosis. So this is where you're pushing your body to produce ketones. So essentially you're starving the body, you're minimizing the exposure of glucose for the body. So this is either a ketogenic diet, which is not just low carb. And I have to always kind of drive that point home. It's not just low carb. It's very, very low carb, as in about 5% of your calories come from carbohydrates, very high fat. And then the rest is protein. So typically about 15% of your calories come from protein, 10 to 15%, and then the rest of that is fat. And so essentially you're pushing your body to where it no longer has that glucose to burn as a fuel source. So it breaks down fat into ketone bodies, which your body then uses those ketones as a fuel source. You can also push yourself into ketosis through fasting. Obviously, especially with the populations that we work with, if you're talking about Navy divers or aviators, military personnel in general, these strategies sometimes aren't necessarily optimal, especially in the field. One, you're having to eat very particular types of food. If you're on the diet, fasting is not ideal for a number of different reasons, especially when it comes to energy expenditure in those individuals. And so DARPA actually funded, so the US agency DARPA actually funded a study to produce a ketone that individuals can consume. So they can eat a normal diet, but essentially they can consume this fuel, this optimal fuel for individuals when they're working in these extreme environments. So that would be exogenous ketosis. So instead of forcing your body to produce ketones to burn as a fuel source, you're giving your body ketones through consumption of ketones. You can also consume things like MCT oil or coconut oil. So you can see on the left, that bumps up your ketones that are available by a little bit. You can also consume ketone salts. So those are all over the market now, and they'll bump up your blood ketones just a little bit more. But these ketone esters that DARPA funded to be produced are actually the kind of the biggest bang for the buck when it comes to optimizing the ketone fuel source. And so we found in studies pretty consistently that the ketone esters that have been produced that we've tested so far will actually consistently and reliably within 30 minutes bump your blood ketones up to about 1.5 to 2 millimolar, and it'll peak out around 3 to 4 millimolar, and then it drops off depending on what your metabolic rate and how active what your metabolic rate is and how active you are. So it's been a really interesting fuel source, and so these ketones are produced, and then the question comes: do they actually have an impact on performance and protection in some of these military relevant environments? So one of my colleagues, Dr. Dom D'Agostino, he's actually a leading researcher when it comes to ketosis, and especially when it comes to neuroprotective strategies and also just diet in general. He did a study, he's kind of thinking about ketones. So in the past, there was a whole history with ketosis, and still is an impact of ketosis in individuals with pediatric epilepsy or childhood epilepsy. And so in those the children that were treatment resistant, they found that putting them into ketosis actually essentially negated their seizures went away. And so he was thinking about this in the setting of this CNS oxygen toxicity in divers that I was telling you about earlier. So this is where you breathe out high pressure oxygen and you have this risk of developing seizures underwater. This is a really big problem. So, Navy kind of sees this as a holy grail area of research. So essentially, if we could find a way to get rid of the risk of CNS oxygen toxicity, you could actually breathe higher pressure oxygen and really significantly reduce the risk of decompression sickness. You could do longer dives. There's a bunch of benefits if you could try to get rid of this risk of CNS oxygen toxicity. Studies prior to have shown that anti-epileptic drugs, there's a whole series of work that's been done over the last few decades, that essentially anti-epileptic drugs have no impact whatsoever on CNS oxygen toxicity seizure risk. And so we're still looking for something that could impact. So he said, if ketones or ketosis has been used in the past for epilepsy, would it potentially have an impact on CNS oxygen toxicity? And long story short, he found, at least in a preclinical model of oxygen toxicity, that it does have a significant impact and actually significantly reduces the risk of CNS oxygen toxicity. So he did a study in rats, and this is one where they pushed the rats. Essentially, they typically develop seizures within about 15 minutes. And these rats were actually pushed out. There's over a 575% increased seizure resistance in these rats when they consumed the ketone ester. This is the BD-ACAC2. And it was incredible. It's to the point where they actually started developing pulmonary oxygen toxicity because they were still under this high pressure of oxygen, but they weren't seizing. So it's pretty incredible work. So now we're testing out, does that impact, do you see that same effect in humans? So Duke University actually just completed a study where they looked at ketogenic diet in individuals and actually got an IRB approved study to push individuals to the point of oxygen toxicity seizures in a chamber environment, very safe setup to make sure that they didn't go underwater or anything like that. And essentially they found that the ketogenic diet did improve resistance to development of seizures in that setting. And so now they looked at the diet, now they're looking at this ketone ester to see if it has the same effect in humans. So it's really exciting work for the undersea medicine community. So then some other questions have come up. We know that when you burn ketones, you have an increased heat output. So the thought was that maybe ketones could be protective in a cold water diving environment. So not so much the brain, but this is thinking more about physical and cognitive performance in this cold water environment for Navy divers and undersea operators. And so just for time's sake, I'm not going to dig too much, but we ended up having a study where we had individuals that were immersed in water. It was fairly cold. Their core body temperature dropped to below 90, typically within about 20 minutes or so. We saw reductions in both cognitive performance and also physical performance in these individuals. When they had ketones on board versus a placebo, they actually perform back at normal baseline. So really exciting. So things like dexterity, they actually maintain their manual dexterity with ketones on board when they had versus when they had placebo on board. So really exciting work, and we're getting ready to publish that study as I speak. So we also looked at ketones for hypoxia mitigation. So the thought is kind of talking about how ketones under a stressful condition burn cleaner than glucose. And so the thought was they could be protective in the setting of low oxygen or hypoxia. And this is a pilot study that we conducted when I was at my last institute. And long story short, we found that individuals, when they consume ketones versus placebo in the setting of hypoxia, they maintained what their normal normoxic cognitive performance would be with ketones on board, which is really exciting work. Interestingly enough, we also saw, and I'm just going to let this run in the background. This is my colleague, Jeff Phillips. He was doing one of the eye tracking tasks, and we had a whole slew of different tasks that we were going through. But interestingly enough, blood oxygen saturation also did not drop to the same extent when individuals had ketones on board versus when they had placebo on board. So there's a follow-on study, and we actually saw exactly the same thing in a follow-on study with a much bigger N. And they're getting ready to publish those results now, but really exciting work, and also some really interesting implications when it comes to working at altitude and working, or even individuals like clinical patients who are in the hypoxic setting for in the neuro ICU, for example. So other big area of research for me, so ketones are one big piece and kind of that metabolic efficiency optimization, but I'm also really interested in looking at systems in the brain that we can tap into that could improve neuroprotection or neuroresilience, as I was talking about. And I really got interested in this newly discovered system called the glymphatic system, or the G lymphatic system in the brain. It was just discovered and published on in 2011, so it's still really new. But essentially, it's a clearance system for the brain. So for those of you not familiar with it, essentially when we sleep, what we've learned is that this glymphatic system peaks clearance. So essentially it clears out all the metabolic waste from the day. And so if you don't get a proper night's sleep, if you think about kind of how foggy you feel, and cognitively you may not completely be there or be intact, we think that one of the reasons that sleep deprivation or not a good night's sleep could have that kind of impact is because the system hasn't efficiently cleared out all the metabolic waste from the day. We also are learning that long range in individuals who have chronic sleep deprivation, and we know that those individuals are at increased risk for neurodegenerative diseases. We know that, or we're learning that, we think it's an impact of this glymphatic system, not just not able to clear things out efficiently. There's also a tie with respect to things like chronic traumatic encephalopathy, which can develop after individuals have head injury. And we're learning that after concussion or even other types of head injury, this system breaks down as well. So it's a really interesting system. And one, we're learning how to image it, but two, we're also learning so what? Can we stimulate it, or can we get it to repair itself if it's not working efficiently? And the reason I got interested in this system was because of what was previously known as there was VIP, there were previously named conditions, but essentially now we call it spaceflight associated neuroocular syndrome, or SANS. We have these changes to the eye and the brain. And the thought was kind of thinking about, from my background as a diver and thinking about fluid shifts in the body, we know that you have fluid shifts in space and you have this headward fluid shift. And the thought is, if you have this brain lymphatic system that drains, and by the way, it drains based on gravity, so it drains from your head down into your peripheral lymphatics. If you have this headward fluid shift, it seems to make sense that it would be backed up and it's probably not clearing out efficiently. That's what we're hypothesizing. We're still doing a lot of work to prove that out. But I was like, maybe this is a system we can tap into for optimizing, even astronaut health or crew health, both short-term and long-term. And this is just a schematic of the glymphatic system. I'm happy to chat with others just for time's sake. I'm not going to dig too much into it. And I talked about this risk of things like chronic traumatic encephalopathy or neurodegenerative diseases if that system isn't working efficiently. So it's a really interesting space to dig into. And we ended up getting funding from the Office of Naval Research Undersea Medicine program to start studying this system under extreme environmental conditions. And so, long story short, one thing we had to do was optimize the imaging strategy. So prior to our study, the gold standard for imaging the glymphatic system was intrathecal contrast MRI, obviously not ideal for a lot of reasons. And so we ended up validating at least intravenous contrast MRI. And I will say we're also now validating non-contrast based imaging approaches, which are validating out, which is really exciting, I think, for the field in general. So the first part of our study was to validate that intravenous approach for imaging these individuals and looking at glymphatic function. Long story short, we found that it did capture, this intravenous approach did capture glymphatic function just as efficiently as intrathecal contrast administration. So that was really exciting. We're getting ready to publish on that soon. And so now we are in the middle of looking at individuals in the settings of hypercapnia, hypoxia, and also hyperoxia. And we're looking to see how those conditions affect how well that glymphatic system functions. And I'll tell you right now, one of the really interesting things that we've seen is in the setting of hypercapnia, that system shuts down. It doesn't work. It doesn't clear things out efficiently. So if you think about astronaut health, for example, that's a really, think about the potential impact, especially for crew who are in a low level, but still hypercapnic environment for an extended period of time. We also are seeing in the setting of hyperoxia, you see like almost a flip. You see this really interesting shift in how the glymphatic system is clearing. In some places it actually clears out faster, and some places it clears out slower, which some of this makes sense, right? We think all of this is tied to things like cerebral blood flow. So it makes sense if you have vasoconstriction or vasodilation that those things are impacting lymphatics, but to actually see it in the imaging studies that we're doing is pretty exciting. And so we have a slew of these studies. We're finishing the study out, and all this will be published in 2023, just by the way. And this down below is actually not updated because we're almost finished with at least 2 of the arms, which we're really excited about. So next steps for finishing out the study. But now, so again, going from undersea to outer space. So we're looking at these in diver relevant conditions, but they also tie to NASA, right? All those different conditions, hypoxia, and hypercapnia, all impact astronaut crew too. So NASA was very interested in kind of getting involved in what we were doing. And so through Dr. Rachel Seidler, who's at the University of Florida, we have a series of studies that have tagged into what we've been doing for the undersea medicine program, and now we're translating over to NASA. So we're doing a dose response CO2 study. So not just one CO2 condition, but we have 3 different CO2 conditions where we're looking at different doses of CO2 to see how those impact the lymphatic system. We're doing head down tilt plus or minus CO2. So doing that simulated microgravity exposure in those individuals. We're also doing a study where we're doing longitudinal neuroimaging of spaceflight crew out to 5 years post-flight. And we're adding in lymphatic imaging now that we have this non-contrast based approach that's being validated right now. So we're really excited about that. And then there's a totally separate study. It's not NASA, but NASA's tracking and also the Translational Research Institute for Space Health are tracking on this study, where we have the imaging optimization happening for those non-contrast based approaches. But we're also looking at the so what. So can you stimulate the lymphatic system in the setting where either individuals are sleep deprived or maybe a hypercapnic condition where the system isn't working efficiently? And so we're really excited about that study and kind of seeing what the potential future implications are of it. So last but not least, I'm going to just jump in a little bit and talk about this experience that I had working with NASA for the NEMO crew exposure. So if you think about testing for extreme environments, it's kind of, you typically have to kind of recreate these different things. If you're in the field, that's one thing, that's fantastic. So this is me working with a crew of individuals on a group of deep sea rebreather divers. For those of you who are familiar, essentially they're doing really, really deep dives for pretty long periods of time. And so we were doing transthoracic echo of those individuals to look for bubbles in the heart. We were doing blood biomarker studies for those individuals after their dives. But still, it's a little messy if you do those field-based studies, right? You can't really control the environments. You can't control the exposures as well as you could in the lab. So then you have the lab-based exposures, but obviously there are always limitations to how relevant those are. You do the best you can to try and make them as relevant as you can to the extreme environmental conditions that the populations that you're studying for are experiencing. And this is also me working in the Hyperbaric Center at Duke University, where we're doing cold immersion for individuals. And we were doing a study looking at sildenafil actually for protection against immersion pulmonary edema in individuals. So you do the best you can to try to recreate in the lab environment so that you can at least try to keep those conditions as reliable and consistent as possible and try to minimize variability. And the space research, it gets even a little bit trickier. So trying to simulate microgravity, we have things like head down tilt, especially the extended head down tilt studies that they do, the bed rest studies that they do over at DLR or ESA in Europe. So we've worked with them. You have studies where you do the parabolic flight, where you get those bursts of microgravity exposure and also high G exposure. You have isolation simulations with HERA, things like HERA or high seas, which are great, but then you don't have the physiological impacts of spaceflight. So you're always just trying to kind of like test. It depends on what you want to test out in these different analog environments. And so NEMO is an analog environment and a test environment that NASA developed back in 2001. And essentially this is NASA's always really good at coming up with clever acronyms. So this stands for NASA Extreme Environment Missions Operations. And it's a project where they have spaceflight crew sometimes join scientists as crew to live anywhere up to about 2 weeks or so, from a week to 2 weeks. And essentially they live undersea. So there's this physiological isolation because you're living in saturation. So essentially your blood and tissues are saturated with nitrogen after about 24 hours. And so after that point, you can't just come up to the surface. You actually have to go through an extended 16-hour decompression to come up safely from that dive. So you have that physiological isolation. You have a psychological isolation, but not so much the same degree as high seas, because we did have interaction with crew that would come back and forth to the habitat to deliver things or take things from us throughout the mission. But then you also have the psychological piece of things too, right? So just knowing that a bad move, I mean, it's not just that, you know, it would affect the crew's schedule. You could actually potentially kill your crew members if you did something stupid. So there's also that component of things that make it space flight like. And so they do this mission for crew training, but they also do it to test out things like software and hardware or experiments or protocol. They're getting ready to go up to space stations. They can kind of work out the kinks of those things before they go up to station. So Nemo happens out of Aquarius Reef Base. This is an undersea habitat and also the shore base system located in the Florida Keys, so that all the way off the southern tip of Florida in the United States. And the crew there is just fantastic. So Nemo's not the only mission that they run. They have scientific missions, they have military missions that they run there. An incredibly professional group of people with commercial diving backgrounds and military diving backgrounds, and hands down is one of the best undersea crews that I've ever had the chance to work with. So I can't say enough great things about them. And so just kind of pointing out where they are again. So Miami, Florida would be like up here. And then the habitat's about 6 miles offshore. And then this is where the shore-based facility is, where they keep all the equipment and monitoring equipment. And during our mission, we had a lot of different scientific projects that were based out of, the scientists were based out of this shore-based facility in the Florida Keys. And then this is what the habitat looks like. So this is the undersea habitat that we were living in. And I'll show you a video in a second so you get a feel for what the inside is like. We are living at the equivalent of about 15 meters of seawater, and the base of the habitats the base of the habitats at about 20 meters of seawater. So not super deep, but again, we're down there long enough that we go into the state of saturation, so we can't, you know, come straight up from the dive. So that's what this is for. This gazebo is a safety habitat. So if anything were to happen, like a fire, we have an escape plan to get out. We would come up under the habitat with tanks, and essentially they would have the crew come out to rescue us and decompress us safely. The story with that gazebo is it also was used for a very long time as the toilet. And so they didn't have the toilet inside the habitat working. The toilet inside the habitat's here now. But for the longest time, this was the toilet. And so this is actually where you go in and out of the habitat when we're doing our EVA or simulated spacewalks or simulated water walks. And so they told us when we were doing our crew training that for using the bathroom, you'd have to bring your mask, your fins, and a swimsuit. I was like, okay, mask and swimsuit makes sense. So you're getting in the water, you're climbing out from the moon pool, and then you're going into this gazebo, but literally you can like grab your hand on the edge of the gazebo and just duck under. It wasn't like a swim anywhere. It's right there. So the fins didn't really make sense. It's like, why would you need fins for that? And it turns out the fish would start to gather and bite because they knew exactly why you were in that habitat. And so they had some previous like bite incidents, I think, that drove them to decide to reinstate the bathroom for our crew instead of using the gazebo. So we were pretty excited that we were going to be able to use the bathroom. We were also told by previous crew members that we weren't true aquanauts if we didn't use the gazebo as a bathroom. So anyway, that's a little side story. And so this is the inside of the habitat. I'm just going to let you watch this. This is crew that ran and Sea Test 2, which is actually a previous NEMA mission. I'm just going to let them chat this through, and you'll notice probably some of these all current astronauts that are in this crew.
Yes. Hey, so Kernagis from JAXA. Good to be back in the wait room.
Kate Rubens, NASA astronaut, and I'm pretty happy to be here.
And I'm Andy from ESA, and we're really excited, all of us, to start the Sea Test mission. And we welcome you to Habitat Aquarius. Let's take a tour. This is the entry port with the moon pool. As you can see down here behind me, this is where we enter and exit the habitats from. This is where we store our diving equipment. This is where we have all of the air connections. And it also serves up as our shower. Next, we get to the entry lock, which serves as an airlock with the 2 slidable doors. It also has a hygiene station where we can brush our teeth. And most importantly, our comm station where we can talk to the divers who are out on EVA, and with Mission Control Center topside. Stepping further into the habitat, we get to the galley where we can prepare our food. We have a small table for eating where Soichi and Kate are sitting. Hey guys. Hi. And all the electronics for controlling the habitat. And last but not least, we have our bunk room with 6 bunks for the 6 people on board Aquarius. There's not a lot of room, as you can see, but plenty for sleeping.
All right, so I love that video, and also I love that view because that's a view that we would have at sleeping at night. You could see, you know, out through the port, like the port, like what was swimming around outside. And this is a picture I took one night of a guest fish that was hanging out, checking us out, and it was just it was a really great memory to, you know, being down there and also kind of combining that undersea background that I had as an exploration diver now with a scientist and doing that for future spaceflight research as well. So just really quick, this is our NEMO crew. So you'll probably recognize, so Megan MacArthur, who I mentioned earlier. So she was a shuttle astronaut, but also since our NEMO mission, she's actually flown. She had a 6-month mission on space station recently. She's also a PhD in oceanography, just an incredible human. She was one of our 2 mission commanders. Our other mission commander was Reed Wiseman. So he's a Navy test pilot. He's currently chief of the astronaut office, and just both of them, I can't say enough amazing things about them as leaders. Dr. Mark Agrifa, so he's a physician. He's done a lot of work with NASA and other space agencies in the past. He's got his hands in a lot of different really interesting things, but he's just this incredible human. He's worked with NEMO for quite some time and was just a really great asset as far as a team member goes. Dr. Noel Dutoy, so he's an engineer who was actually another undersea folks. So he was working on human machine teaming. So looking at diver / underwater robot teaming in the water. So he's doing that work for the Navy, but he was doing it during the NEMO mission. So again, kind of cool to see that crossover from undersea to outer space. And then Dr. Matthias Maurer, so he's also a flown astronaut now. He was training at the time. He was a very new astronaut at the time, and just another great human to work with as far as crew goes. So they try to make NEMO as similar to a spaceflight as possible. So essentially, this is mission control. It's a makeshift mission control that's put together back at that shore base that I talked about in the Florida Keys. So every day you go through mission briefing and you'd have all the research teams, all the operational teams on site to talk through the next day's activities and make sure everybody's getting the research done, all the tasks are being accomplished by the crew. And you can see up on the screen, this is actually a crew schedule. And one of the things that, if you talk to the astronauts, they say is probably one of the more stressful things on space station is that you're always living by this timeline, except for this little chunks of time where you're given off. But when you're on this timeline, it's a tight timeline because they have things scheduled, not just so you can get your work done, but so your colleagues can get their work done and you're not stepping on each other's toes. So each of these blocks is a different task. These different lines denote, so the red line is where you're actually at during that, at that time specifically. And these dotted lines actually simulate mission time delays. And so during our mission, we actually had a mission time delay based off of for communication because they wanted to simulate that for longer duration flights in the future. So that was always something you kind of kept in the back of your mind from morning till like late evening. You were always on this timeline getting your work done. We also, another thing that made it similar to spaceflight is you had intravehicular communications. So we had individuals who are out doing EVA or extravehicular activity. So this is kind of like doing a spacewalk, but it was out on the ocean floor. And so we would communicate back to the person who is on IVA or IV, and he would communicate back to mission control. And by the way, there's a time delay as well with that communication loop. So they're trying to make it, again, as similar as possible as we're going through these different activities during the mission. There are a lot of different projects. So over the course of a 2-week mission, a 16-day mission, these were all the different projects that our crew was working on. So prior to the mission, we did a lot of training to make sure we were familiar with a different protocol before we got into the habitat itself. But lots of different projects. And it wasn't just NASA projects, by the way. We're doing work for the Navy. So we had undersea medicine projects taking place. We had private industry projects taking place. We had projects that are funded by National Science Foundation. It was a really interesting slew of different projects that we were doing and trying to maximize the crew time and also just the exposure that we were getting during the NEMO mission. So just some examples of projects. So this is actually a medical telemetry study where we would capture data every day, morning and night, and have that relayed back to a medical team on the surface. Obvious implications for future space flight. This was a study where we were testing out equipment that would measure vestibular function and balance. And so we would do a series of different activities wearing these devices. And it was interesting because you see Megan doing the arms cross, eye open task, and then we would have one where we close our eyes. And one where we close our eyes, we would often veer off into the refrigerator, and it's because the habitat actually had a tilt that we found out about later. So that was kind of funny. It's like, why do we keep hitting the refrigerator? But I thought that was interesting. Probably one of the fun, more competitive ones, internally competitive in a great way, was working with the MinION device. So this is a device that eventually, taking the protocol that we tested in NEMO, went up to space station, and Kate Rubins was the first person to sequence DNA in space using this device and the protocol that they had developed. So we were testing out the protocol for them, just seeing, if there are any hiccups or kinks or anything like that, if we could successfully extract DNA from samples that we swabbed in the habitat. It ended up becoming a little competitive to see who could get the first successful DNA sequencing, but also the most productive sequencing too. So it was a kind of a fun project for us to work on. We also tested out the miniature exercise device 2.0. So the idea with this device is you do exercise, but you're also tagged back in. The idea for NASA is that you're tagged back in with somebody on Earth, so you could do joint exercise or shared exercise with a family member, for example, or another crew member, or somebody along those lines. So you have that social interaction combined with exercise. And so we were testing out how well that program worked. So those are just examples of the projects we worked on for time's sake, but I'm happy to talk about anything else that we did there. For Splash Up, so essentially we were in this habitat that was opened out to the sea. So we were equilibrated at pressure during the entire mission. When we're getting ready to decompress, they closed that hatch to the habitat and slowly overnight decompressed us. So essentially we went from being this open sea habitat where we're open to the equivalent pressure at depth, to now we're after that 16-hour decompression, we were like a submarine. So we're at 1 atmosphere equivalent sitting on the bottom of the ocean. And so this picture on the left, everybody does an oxygen pre-breathe. So it kind of helps expedite getting that nitrogen out of your system a little bit faster before you start that decompression process. And everybody's reading 20,000 Leagues Under the Sea, by the way. We all had a copy of 20,000 Leagues Under the Sea that the crew all signed for each other and brought back up after the mission. But anyway, so after you're at that equivalent pressure, we were checked by a physician to make sure that everybody's okay, no signs of decompression sickness. And then they repressurized the habitat, opened it back up. So now you're equivalent at the equivalent pressure again. And we put scuba equipment on and swam out of the habitat back up to the surface. And that was how we ended the mission. Just by the way, I mentioned just kind of this combination of what I did before as my career and now my second career in research. This is probably that MinION device is really fun because for my PhD work, I did a lot of work with RNA and DNA sequencing, many late nights, overnights, sitting in the lab sequencing and extracting samples and sequencing RNA and DNA. And I will say that I love this project because it really tied together that undersea exploration background that I had into the current research career I had. So I sent a picture to one of my PhD advisors and I said, just by the way, sequencing DNA undersea is way more fun than it is sitting in the lab at like 2 o'clock in the morning, excuse me, sorry, cough. So anyway, really cool experience and just a great way to kind of tie things together. So I just want to give a shout out to my crew, all my previous lab members, my current lab members, and all the funding agencies that have supported the work that we've been doing, and obviously the NEMO staff and crew as well. So with that, I'm going to let this video run in the background. This is a video of world record breaking free dive. So this is a breath hold dive. So a little bit different from the scuba diving I was talking about. But it's just absolutely incredible to see what humans can do and like where they're pushing their capacities. So I'm just going to let this run in the background. I'm happy to answer any questions about this free dive too as it goes. So with that, I'm done, and it's really great to get a chance to chat with you all today.
Thank you so much. Out of curiosity, how far did they dive while breath holding?
So this dive is 100, and I want to say it's 100. It's going to say on the bottom, I think it's 155 meters. It's pretty insane on a single breath, down and back, no weights, just swimming under their own power. I want to say that I want to make sure I have that right. 130 meters, sorry, 130 meters. Yeah, only 130 meters, but no, it's absolutely incredible. So Alexei Molchanov is absolutely one of the best freedivers in the world. So it's really cool to watch him and see what he's able to do. So
I don't think I've ever personally done a dive that was deeper than 100 meters with gear.
yeah, yeah,
Like with tanks. That's crazy.
yeah.
Oh, right. Well, in the interest of time, let's let this play and start going through the questions. I'm happy to do my question last. Melissa, would you like to go through the questions while the video is running? And we will stop recording now.